Hello guys, welcome back to my lesson. So today we're going to answer questions under the topic of DNA code of life. Let's look at the first question. It says the diagram below shows part of a process involved in the production of a protein. So if you look at this diagram, it is showing the ribosome, it's showing transfer RNA and mRNA. So this is transcription. Identify molecule Y. Molecule Y is tRNA. The group of nitrogenous bases at Z, that is anticodon. And then it says if X is the next amino acid required after W, then identify nitrogenous bases 1, 2, 3. So if X is after W, then it's going to pair with the nitrogenous bases A, C, C. To find 1, 2, 3, you're going to use the sequence DNA. Under DNA, mRNA. Under mRNA, you write tRNA, and then you substitute mRNA A, C, C. A pairs with U, C pairs with G, C pairs with G. So the answer is UGG. The next question says the DNA-based triplet that codes for X. So now we have we have mRNA and then we have tRNA. You can choose either of the two to find the DNA, but let's go to the sequence again. DNA, mRNA, tRNA. You substitute, let's say you substitute the tRNA, which is UGG, right? So UGG, you're going to move from tRNA to mRNA. U pairs with A, G pairs with C, G pairs with C. mRNA is ACC. And then you move to DNA. A pairs with T, C pairs with G, C pairs with G. The answer is TTG. So let's look at the next set of questions. It says that the diagram below shows the process of protein synthesis. As you can see that there is the nucleus there, and then we have the ribosome, we have the mRNA and the tRNA. And the question says that state the two organelles in the cell that are directly involved in the process of protein synthesis. We have the nucleus and the ribosome. How many codons are present in molecule Y? How we count codons is that you count 3 nitrogenous bases, that is the first codon. AUG, that is the first codon. CCA, the second codon. AGA, the third codon. So the answer is 3. Name the bond represented by T, that is the peptide bond, because it is the bond between the amino acids. Give the antigon represented at Z. So the first vertical small lines there is AUG, and then at the top of 1, 2, 3, it's going to be GCC on the mRNA. So we have to find the tRNA 1, 2, 3. Those nitrogenous bases, we're going to use the sequence DNA under DNA mRNA. Under mRNA, tRNA, we substitute. On the mRNA, we have CCA, right? So CCA, we're moving to tRNA. The answer is GGU. So let's look at the last set of questions. It says that 4 soldiers are missing from an army unit. The unit finds the body of one of the soldiers that is missing. And then they have been killed in an explosion. There are no identifiable features on the body. And then the identification tags are also missing. So the army asks the parents of the 4 missing soldiers for a DNA sample so they can compare the DNA profiles of the parents with the DNA profile of the soldier that was killed. So you're going to look at the DNA bands of the soldier compared with all the parents. Now let's start with parent A and B. The first band matches with no one. Second band matches with both of them. The next band matches with nobody. The other band matches with nobody. Another one, it matches with parent B. Another one matches with parent A, parent A, parent A. And then the last 3, only 2 of them matches with parent B. Let's go to the next parents. The first band matches with parent D, the second one parent C, third one C, fourth one D, fifth one D. Another one matches with parent C, the next one matches with parent D, another one D, another one D, and then another one matches with parent C. So there is no need to compare the DNA bands of the soldier with parent E, F, G, H, because if you look at all the DNA bands in the soldier are found in parent C and D. So it means the parents are C and D. But if you want to compare them, you can continue and compare with those of parent E and F. So the first question says, which set of parents are the parents of the dead soldier? The answer is C and D. Explain your answer because the DNA bands found in the soldier are all found in parent C and D. Mention one other use of DNA profiling. It is used for identifications or identification of criminals, identifications of genetic disorders, developing cures for genetic disorders, tissue type for organ transplant. Thank you so much for listening. Don't forget to subscribe to the channel.